Engineering. So we are called an institute. So we're actually a department of biomedical engineering, but we are called an institute at the university. The reason is that we're actually under 3 faculty. So faculty of applied science and engineering, faculty of dentistry, and faculty of medicine, while all the other engineering departments are under the faculty of applied science and engineering only. So because we're under 3 different faculty, we're called an institute within the university's governance. But because we're under 3 different faculty, the institute actually has students from different backgrounds: chemistry, biology, engineering, psychology, and I think there's been a few students that have been in law school, right? That's entered into the graduate program. So lots of diversity and flexibility in terms of the program. There's 40 main faculty in the institute, and we also have 60 cross-appointed faculty that are from the hospital. So they go through us for students and for teaching. As most of you know, the institute is in the downtown campus, and we're also affiliated with 7 different hospitals. There are approximately 350 graduate students in the institute. So as you know, we don't have an undergrad biomedical engineering program at the University of Toronto. So our main program is really with graduate education. We do have a minors program. We do have an option through engineering science. There are essentially 3 major research themes: molecular engineering, cell tissue engineering, clinical engineering. Molecular engineering dealing with molecules and structures to create new therapies using molecules. Cell and tissue engineering uses cell and tissue to regenerate tissues to improve the quality of tissues for therapeutic and diagnostic purposes. And clinical engineering tends to work with patients. So a lot of researchers develop things like braces, new ways, pacemakers, all these kind of things that are more directly working with patients. So the institute have a broad range from basic all the way to applied, working with molecules all the way to patients. Please check out the website. The one that might be most relevant is this discover.bme.utoronto.ca website, which gives stories and editorials about life in institute as well as research activities in the institute. The more administrative website is bme.utoronto.ca, like how to apply to the program, what to do as a student, and so on. Right? So please feel free to ask us about the institute. So I'm going to talk a little bit about my research program. So what my research program is interested in doing is to develop nanoparticles to target cancer. So we as engineers can build nanoparticles that can image tissues or can cure drugs. So we develop these particles outside the body, and we can inject it inside the body, and they circulate around inside the body. And they will eventually accumulate in the tumor. And there are 2 reasons why they accumulate in the tumor. One is that as the tumor starts to grow, there's little holes that form in the vessel. These holes tend to be 200 nanometers upwards to 1,000 nanometers. So things that are smaller can diffuse through these holes to get into the tumor. And at the tumor, you can release the drug and kill the tumor cells. The second approach is that when the nanoparticles are moving around the body, it picks up proteins, and those proteins can recognize specific receptors on the blood vessels, on the tumor blood vessels, and that can also carry it into the tumor. And then again, you release a drug and you can essentially kill the tumor that way. So the reason everyone's excited about these kind of cancer nanotherapies is that the nanoparticle can store lots of drugs, thousands of drug molecules inside one nanoparticle. So the theory is that because it stores so many nanoparticles, you need very few nanoparticles to get in sight. So you can actually have improved therapeutic effect to the patient. And it can also reduce side effects. One of the reasons it can reduce side effects is because the drug is trapped inside the particles and it doesn't actually interact with the healthy tissues. And the reason you get side effects is because the drug starts to interact with the kidney, the liver, and all these organs that may not have a tumor, and then it actually causes problems and you express the side effects. So this is the main concept around cancer nanoparticles. However, what we've been doing, our lab and many others have been actually measuring the journey of a nanoparticle, right? So you have a particle outside, you inject it in the body, it's moving along the bloodstream. And what ends up happening is that inside your body contains a lot of cells and tissues and molecules that actually try to trap the nanoparticle and prevent it from going to the tumor site. And so in the last few years, we've actually measured how many nanoparticles goes into the final tumor site. And so this is just a rough number. So if you inject 1 million nanoparticle, what ends up happening, 7,000 of the nanoparticles will reach the tumor as a whole, while the other 993,000 will go into other organs like the liver, spleen, bone marrow, and other organs. And if you try to target the tumor cells within the tumor, only 14 of those million gets there. And if you try the nucleus of the tumor cells, only one of them gets to that site. So the objective of my research program is to develop strategies to increase these numbers, go from 1 million to, let's say, 100,000, which then increase the amount of tumor cells, which increases the number of nucleus, which in effect should lead to better therapeutics. So we're trying to create a better targeting system, and that's sort of the main concept. The philosophy in my lab right now is that if nanoparticles are not delivered into the targeted disease site with a high enough dose, they cannot work as designed. So we're trying to increase this number through design of the particle and through understanding of the biology. And so one of the big strategies we've been trying to do this is by using a computational technique. So what that means is that we've been designing different types of nanoparticles. We've been injecting it into mouse models with different kind of disease characteristics. And then we can also look at the mouse's characteristics, such as age, sex or gender, the history of the mouse. So you can create different kind of problems with the mouse. So you can look at how the particles respond to these particular parameters. And then you can measure how the particles relate to how the particles behave inside the animal, such as biodistribution. How does particle design affect how they distribute in the body? If you change the size, the shape, the surface chemistry. How do they distribute with the different disease characteristics? So the tumor's early or late, small or big. How does that affect it? So those are output signal. And then what you can do is you can use computational analysis to correlate the input with the output. So you can use anywhere from decision matrix and linear regression, which is a very easy technique, to using machine learning, which looks at multiple variables and how they relate to these parameters. And so if we can develop this algorithm, it will allow us to now go through this flow of analysis. So our future, the way we think about nanomedicine is the following. A patient goes to a hospital and gets a imaging or a pathology analysis to be able to characterize the disease. That information feeds into the algorithm that you created. That algorithm will tell the engineer, the chemist, to make the nanoparticle of a specific design. Then you administer it to a patient, you monitor the effect, and then you repeat this process. If the process does not work, then what you need to do is create an adjuvant. So what that means is that you have to add something that affects some of the cells and tissues that take up nanoparticles, so more of them deliver into the patient. And then you basically administer and you repeat this process. So what we're really doing, the bigger picture, is that we're building technologies to personalize the treatment for patients with cancer, right? So for example, a patient at stage 4 may require a 50 nanometer nanoparticle. But if the therapy works well, the next time they come into the hospital, they might need a 20 nanometer particle because the biology of the tumor changed through time. So you actually develop this algorithm to allow you to determine what's the best design for that particular therapeutic application. And the bigger idea we think about is the nanoparticle essentially is a delivery system, right? It's a carrier to transport a drug from point A to B. And if you look at the world we live in, there's a boat for going on water, this plane goes on the sky, there's cars that's going on the road. So the question that we've been asking now is, what is the best nanoparticle carrier design to deliver therapeutics to solid tumors, right? So you custom design the delivery vehicle based on the terrain, and we're trying to figure out how to actually design the particles for the terrain in the body. And that's really what one of the things we're focused on at this point. And as you can see, we're very good at making particles in this point. So you can make them different shapes. You can make them different sizes very easily. We can also make nanoparticles from the biological fluid. So in this example, we can actually take blood from a human patient, coat it onto a metal particle, remove the metal nanoparticle, put a drug inside, and then re-inject that into a patient. So you can actually use the blood's own biology to be able to build your nanoparticle system. So we have a lot of different ways to make them. We're also now developing different imaging technologies to be able to analyze the tissues. So here's an example of a technique we developed where tissues are made optically transparent. And then we label them with different colored dyes that can label different molecules and structures within the tissues. And the reason that we make these tissues clear is because of the fact that light can penetrate into a tissue when it's transparent as compared to when it's not. You can do organs, you can do whole animals. Here's a whole mouse that we made optically transparent. And here's an example of nanoparticles that are highlighted in red with vessels that we essentially label. So this is a tumor. And you can clearly see how the nanoparticle delivery vehicle is surrounding the vessels, where they're located at. Certain regions have more nanoparticles than others. And the point of this is to be able to take human biopsy samples and analyze it in 3 dimensions. So you can actually see all the molecules being produced, all the cells that are being produced or killed, and be able to use that as a way to guide the design of your nanoparticle formulation. So the bigger issue here is that we need to develop better delivery system, right? So if you look at your body as like a big highway system with different entry and exit ports, your point is that you're trying to make a drug to go to a specific port. All the new therapies that's been created the last 20 years require delivery to a specific target site. Genome editing, synthetic genomics, a lot of small molecule agents, immunotherapy, molecular machines, engineered proteins. So they require delivery to the target site for them to work as designed. So the conclusion I have in this short presentation is that there's a lot of exciting research going on. We're still trying to figure out what cells and tissues are the barriers that block the targeting of nanoparticles. And we're learning how these cells and tissues affect nanoparticle design. And what this kind of area of research is called the nano-bio interactions. And what it does is it sets up the foundation for chemically designing nanomaterials for in vivo cancer applications, as well as other diseases, heart disease, diabetes. They all require you to take a drug from point A to B. And we're just trying to figure out how to do that at this particular point. Here's my group. It's very diverse of people from very different backgrounds. Lots of engineers, lots of biologists, lots of clinicians as well that have joined my lab. So I'm not going to go through their names, but they are very integrated in terms of work. And we actually collaborate with a lot of people from in Toronto, outside Toronto. These are Toronto folks. This is outside Toronto to actually do these projects. And we are being very well supported from engineering councils as well as health agencies in terms of our work. It crosses 2 different areas. Thank you again. And I'll look forward to see the next professor's talk.
Thank you very much for your presentation. I really liked your comparison with the vehicles and stuff. That was really interesting. Now I'm going to pass it over to Lidan Yu. She is the professor of the Mechanical Engineering and Biomedical Engineering departments at UofT. And so I will pass it off to her.
Okay, let me share my slides. Okay. Oops. Okay. So can you see my screen?
Yes.
Okay. Yeah. So hi everyone. It's really nice to be here and share with you our research. So today my topic will be developing microfluidic chip systems, mechanobiology on bone on a chip for bone mechanobiology study. So first, this is my lab, and we have, again, just like Professor Chen mentioned, we have students from all different kind of background, from mechanical engineering, chemical engineering, material science, molecular biology, immunology. So it's really very dynamic, very integrated, interdisciplinary team. So why we're interested in bone? So bone is basically a structure that most people think is a solid and kind of a static, but it's actually a very dynamic structure, and it's actually a very portal structure, has a lot of pores and filled with interstitial fluid. So our bone is just like a muscle. We can adapt, our bone can adapt to mechanical environment. For example, what's shown here is the bone in the playing arm of tennis player. As you can see, it's much denser, much wider compared to the non-playing arm bone, right? So suggesting the bone can adapt its structure to meet the mechanical environment. Now, if you're an astronaut going through a trip to Mars, you will heavily lose your bone like crazy. So basically it's like a 3% loss of bone every month, right? So basically that means if you don't put a lot of mechanical loading, right? If you're doing bed rest or space travel, so you will have a problem, we call it osteoporosis, which means there's a lot of pores created in your bone, right? So bone is really a smart structure. Basically, it tells our bodies experiencing mechanical loading and will tell our bone, okay, now is the time to get more bone so we can do more exercise. Or if that person is lazy just staying in the couch all the time, then you don't need a lot of very strong structure to support your body, right? So then your body will just order the bone to lose more calcium, to lose more bone, right? Okay, so in our lab, we are interested in the general, the big question we ask is how mechanical load can actually affect changes in terms of structure changes in bone. So specifically, the recent years, we're focusing our efforts on the last two questions is how does mechanical loading affect intercell population crosstalk and how that regulate other diseases? And one of the diseases I'm going to talk about today is bone metastasis, which is breast cancer bone metastasis. And also we ask, can we develop a better in vitro platform to study bone cell mechanical transduction? So before we go into details of that, so I just want to give you a very brief introduction of what's going on in bone in terms of cell population. So in the bone, right, it's just like a huge building that has many apartments. Each apartment, you have one cell living in it. It's called osteocyte. These cells are composing 90 to 90%, 95% of all bone cells in our bone. Now, there's also osteoblasts, which are builders. They're responsible for bone formation. And there's also osteoclasts, which is responsible for bone resorption. And then there's also stromal cells, which can actually differentiate into either builders or, into osteoblasts. So now osteocyte, which I just mentioned, is the residence of these big buildings, right? So they are actually very sensitive to mechanical loading, which I'm going to show two short video clips, as you can see. So basically what we do is we apply 1 Pascal dynamic bidirectional shear stress, free flow shear stress, and then look at how the cells respond to this mechanical stimuli. On the left-hand side, this is actually 1 Pascal static flow. So the flow direction is from left to right. And this is actually 8 hours video compressed to less than 1 minute. So as you can see, the cells are moving around, but quite laid back, I would say. Now, if you look at the same cells experiencing dynamic loading, which is bidirectional, so the flow goes to here and then back. So it's a 1 Hertz frequency, and also at a peak shear stress level of 1 Pascal. So in terms of the magnitude, these two are the same. But look at how cell responds, right? So the cell actually responds very differently. They try to reach out to each other with these protrusions, build up connections, just like our human society, right? They're trying to build a community here. They try to crosstalk to each other and very active. So that shows you how active this osteocyte, how sensitive is this osteocyte in response to mechanical loading. And the cells can respond with intracellular calcium. What's shown here is actually a video clip showing the increase in terms of intracellular calcium concentration. Now, intracellular calcium increase is an important second messenger that will actually lead into a lot of changes in the downstream signaling pathway, which in the end can lead a lot of changes in gene level expression. So as you can see here, the color, the pink color shows, just like fireworks, shows the high concentration of intracellular calcium. Green and bluish means the baseline calcium concentration. So when we apply free flow, as you can see, the cell instantaneously responds with calcium increase, right? So in our the platform we're trying to develop, right? So we used to have used the parallel plate flow chamber, which is very simple, just the two plates with the gap in between, and we have cells grows on one of the plates, and then we can deliver flow. The cell can experiencing shear stress between the two plates. The problem with this approach is that it's a lack of half-life signaling interactions with other cell population. There's no direct communication between different cell population. And also you need a lot of cells, so large cell numbers required. So if we want to test certain drugs on patients, on some cells, right? So then probably we're not going to have that many cells. So then this approach will be limited. So in our lab, we try to develop this microfluid approach. We can have multiple channels with different cells sitting in channels, kind of have crosstalk between different cell population. Each cell can have its well-controlled mechanical environment in terms of shear stress, in terms of free flow. By the way, maybe I forgot to mention, so why free flow, right? So if you think about the bone, most people think it's just a solid structure, right? But I mentioned this solid structure is actually filled with interstitial fluid. So when you apply loading to the bone, what happens, just like a sponge, but it's very rigid, stiff sponge, soaked with water. So you apply loading, you kind of squeeze the water out of the sponge, and then you release. And then the water comes back, right? So I'm just using this analogy to show what's going on, right? So, so that's why we apply free flow to simulate what's going on in the in vivo to the bone cells. And we develop a multiple, like a co-culture system, so we can have one cell population cultured on one channel with other cell population cultured on the other channel, and they can crosstalk to each other, and we can apply different shear stress. We can look at the cell, certain markers expression using immunofluorescence assay, or we can actually collect the cells later and look at gene expression changes. So that's on the platform side. So we're trying to using this microfluid platform to understand how cell responds to mechanical loading, how cells will actually crosstalk with other type of cells. Now, the second project I want to talk about, which is the recent focus of in our lab, is how we use this platform to study cancer bone metastasis. So the cancer we're looking at is actually breast cancer. So why breast cancer bone metastasis? Well, bone is actually the major site for metastasis for breast cancer, right? So it's a metastasis means the spread of cancer cells. 85% of advanced breast cancer patients actually have this problem, and there's no cure about that. The only way you can do is prevent or prolong, right? So to delay the progress. So the progress of the disease. So the loading, everyone knows mechanical loading is good for your heart, good for muscle, and there's also extensive studies from our lab and from other labs as well shows that loading good for bone. Now, the loading from active exercise can actually having a lot of impact, but on the bone, basically, it will promote bone formation and prevent bone resorption. Interestingly, resorption, the cell osteoclast, which is the bone resorption cell, can actually have very active crosstalk with cancer cells. So when you have bone resorbed, the matrix will release a lot of proteins trapped in the bone matrix. And these proteins actually serve as a signal, kind of like telling the cancer, okay, this is the site, you need to come over here, okay? So they serve as the postcode. So the cancer cell somehow can actually sense this and then come all the way to the bone site and start to crosstalk with osteoclast, which is the bone resorption cell. And then this is a vicious cycle, and then you will have a tumor growing in the bone, right? So your bone will be damaged. So mechanical loading actually can tell the osteocyte, okay, now is the time to build more bone to stop bone resorption. So in a certain way, it will basically stop this chain of reaction, stop this path basically. So our hypothesis is that when the osteocyte experiences mechanical loading, it can crosstalk with osteoclast, it can also crosstalk with osteoblast, and also endothelial cell, which are the cells lining on the blood vessels. It's the first line in contact with tumor cells circulating in the blood vessel, right? So we have done extensive studies showing that osteocyte under mechanical loading can actually promote osteoblast formation, prevent osteoclast formation, and stop cancer cell proliferation, and also can make endothelial cells less sticky, so the tumor cell won't be able to or easily attach to the endothelial cell and start this activation process. So we built our microfluid device where we can actually make these artificial vessels, and then we can have bone cells here, and we can have these vessels lined with a HUVEC, which is a cell line to model endothelial cells. We can have tumor cells injected to it, and then we can actually real-time tracking the invasion or the real-time tracking the migration of cancer cells, which is dyed in green, to going through the side channels towards the channels where osteocytes lived in, right? So what we found is that when we apply fluid flow, right, this migration is dramatically inhibited. Right? So, and we, oh, unfortunately, I forgot to include that. This is just the confocal imaging of the blood vessel we stained with weak adherine, which is the proteins expressed on the endothelial cells. So we show that when the flow has, when we apply flow on bone cells, right, and this is a flow on cells, this is no flow, we show that a huge decrease in terms of activation distance, which basically is for the cancer cells going through this endothelial layer and then going through this hydrogel to through the side channels. So we measure the distance. We also measure the percentage of how many cells get across. We found that in both groups, we show in both parameters, we show this decrease when the flow is applied on, right? So I guess that's yeah, that's the end of my presentation. So we're very excited right now. We're actually in collaboration with our collaborator in University of Delaware and working on the animal model, and also in the future we want to test on the patients. So we're actually applying another type of mechanical loading to the animal, and we actually have some very exciting data shows that indeed the mice and their mechanical loading has much less tumor burden, right, growing in the bone. Right? So yeah, I look forward to continue to talk to you in the later sessions. Thank you. Okay.
Okay, thank you so much, Professor Yu and Professor Chan. Now I think we'll move into the breakout room portion of tonight. We do have Professor Khan's presentation left, but oh, there. Hello, Professor.
Okay, if you don't mind, I can start now.
Yeah, for sure. Go ahead. I think
Great.
you should be able to share now.
All right. Let's start sharing. All right, everybody. Thanks for the opportunity to join in tonight. This is a lot of fun. Sorry, it took a bit longer to put the kids to bed than I thought it would, but I'd like to talk to you today about some of the work we're doing in my lab. We focus on multiplexed nanoparticles for multi-gene editing. Let's start off to tell you a bit about my team. We're a new but growing lab at BME at UofT. Right now we have 5 people: Anson, Julian, Alana, Grayson, and Janice. And there are blue coats. We wear fireproof lab coats that are made out of nomics, blue coats. So we do a lot of chemistry as well as biology. All right, so our work, we focus on multiplexed nanoparticles that enable multi-gene control in cells. The idea behind this is that we build RNA nanotechnology platforms that simultaneously control the expression, suppression, and regulation of any combination of disease-related genes. And the idea is that with just a single particle, you can put in different types of payloads, in this case, different types of RNA. And when they get into a cell, they coordinate multiple things at once. So some genes might get turned on, some genes might get turned off, others get regulated, and then you expect some kind of phenotypic change in the cell. And that's the type of work we do. a lot of the stuff that's out there right now only focuses on one type of RNA at a time, and they do one thing. But why are we interested in looking at multifunctional, multiplex nanoparticles? Well, the reason is because diseases are very complex, and they're often caused by several aberrant mechanisms of actions or genes. Now, we're fortunate because we have part of the tools we need to address this. We have these different types of nucleic acids, ribonucleic acids, RNAs. If you use an mRNA, for example, you can express a gene. A small interfering RNA, an siRNA, you can silence genes. MicroRNA regulates genes. A guide RNA can be used with gene editors to guide genetic edits. And there are also replicons, which are self-replicating mRNA. So with this combination of tools, we can potentially achieve multigene control. But the trick to making that work really well is how you deliver those RNA. You need the best possible delivery materials to do this. And it's not just about packing all of these things into a single nanoparticle, which in itself is not trivial, but it's also related to evolution. Now, your delivery materials that you use to collect your RNA and make them into nanoparticle form, they must not diminish the RNA's efficacy. So here is what can happen. Throughout our millions and millions of years of evolution, we have developed these germline encoded pattern recognition receptors in us. And they are designed to recognize common elements in pathogens that, once recognized, will set up this pathway and cascade inside your cells to protect you, because it's worried that you're going to be hijacked by some pathogen and become sick. So your nanoparticles, the materials you use to make your nanoparticles, can inadvertently trigger these receptors. And let's look at what happens when you trigger those receptors. You have a cascade of events, and then ultimately you can get inflammation and stalled translation, which is not very good if you're trying to deliver mRNA and you want it translated. Inhibition of mRNA replication, not so great if you're trying to deliver replicons or self-replicating mRNA, and even RNA degradation. If you degrade your RNA payload, you won't even get a chance to use it. So it's important that you design the molecular structure of your delivery molecules that are used to combine with the RNA to form these nanoparticles. So we work on designing these better molecules. So we work on a molecular level, designing the right functional groups and the right structure to go on these so that they do very well at not only delivering a plethora of different RNA inside a single nanoparticle, but also make sure they don't trigger any of those deleterious pathways. And if you can solve this, you can do some really interesting biology. And I'll give you a little highlight of some stuff that we have looked at in the past that highlights the need of multiplex or multifunctional gene control. Let's take, for example, myocardial infarction. When this happens, you have damage to the heart and you have inflammation in the heart tissue. And what happens here is that you get an influx of monocytes. Usually monocytes go to the site of injury. They help clear out debris, clean out the area, help you recover better. But for some reason in myocardial infarction, you get too many monocytes. And when they get inside the heart tissue, they actually increase the amount of inflammation, bring in more monocytes, and they actually do this strange repair job where they just make a bunch of scar tissue. Scar tissue in the heart is terrible. It doesn't pump blood. It's non-functional. It's dead weight. So you really want to actually control the number of monocytes going into the heart after myocardial infarction. So here's a picture of some heart tissue. And this is a control animal that had a myocardial infarction. And what we did is that we looked at the scar tissue. So pink is normal, healthy, and blue is the amount of scar tissue that forms. So you can see after myocardial infarction, there's lots of scar. Here with treatment, we actually added in a nanoparticle that delivered a small interfering RNA that silenced MCP1. MCP1 essentially shuts the door to monocytes exiting their site of origin, the bone marrow, getting inside the blood, and making it to the heart. So we literally close the door in the bone marrow so the monocytes can't leave. And when you do that, you actually get a lot less scar formation. So here is the graphical version of that. Here's a control. You see a lot of scar tissue formation. But when you close the door in the bone marrow with one siRNA, look at that. You just reduce the amount of scar tissue, which is excellent. But as engineers, we have to look at the whole picture, that entire system. So if we look at the number of monocytes in the heart tissue, so here's the control, no treatment, there's a lot of monocytes. And when you shut the door in the bone marrow with that small interfering RNA, you see that we get less monocytes in the heart tissue. Makes sense. We saw less scar tissue. Where are the monocytes going? So we go back to the bone marrow and we look. With control, with no treatment, all of the monocytes are being generated and they're exiting the bone marrow, no problem. So you see a low number. But with treatment, when we close the door, the monocytes, they're still being generated. They are not able to escape, so you build them up. You have a buildup in the bone marrow. And this is the argument for needing more comprehensive control out of your nanotechnology, where we want to deliver multiple types of nucleic acids. So not only are we looking at the bone marrow, so here's the bone marrow, this is where your monocytes are generated, and here's a blood vessel, and your monocytes are trying to exit out of the bone marrow into the blood vessels and travel, go to the heart, for example. So not only do we want to shut this door to prevent them from exiting, but we also want to send signals to slow down the proliferation of monocytes so we don't get that buildup in the bone marrow. So the nanoparticle would be multifunctional. We would close the door. We would tell the bone marrow progenitor cells to stop proliferating temporarily. And of course, we want all of this to be an on-demand therapy because you never want to permanently shut down any of these pathways. They'll just compromise your wound healing and your immune system in general. So we want to just have it as an on-demand therapy where you deliver it to somebody and then it shuts it down. So another thing we look at is we know that we can do great work with mRNA vaccines. We saw it with COVID, they do a great job, but we also look for alternative ways to use nanotechnology to fight viral infections. And one of the things that we're doing is we're making these special gene editing nanoparticles. Instead of editing genes in people, they edit the genes in the virus. So they have guide RNA and CRISPR-Cas9, and the guide RNA actually recognizes genes in the virus. So when you have a virus stuck inside the person, you can then edit that virus by deleting its essential genes. And when you delete its essential genes, it's no longer viable and it can't reproduce, replicate. So a different take on RNA nanotechnology that help treat viral infections. We also look at another type of vaccine. Normally with a vaccine that you use mRNA, you inject your nanoparticle and then you recruit helper T cells like CD4 and CD8 T cells that help give you cellular immunity. So cytotoxic T cells that clear actively infected cells, so get them out of there. And also they help make antibodies. So that gives you this type of protection where the antibodies stick to the pathogen, inactivates it, it's cleared by the immune system. That's one thing that the immune system does. There are actually other types of T cells in the body. One of the T cells that we're interested in are regulatory T cells. So we have these naive regulatory T cells that actually recognize your tissues, self tissues. And if they recognize your tissue, they tell all the other immune system cells, don't attack this tissue, it's one of us. When that mechanism fails, you get autoimmune disease because your immune system attacks your own cells. So we also make nanoparticles that prime regulatory T cells that are naive so that they can better communicate with the rest of the immune system that this is self tissue, let's do not destroy this. And that's very important because many diseases from arthritis to multiple sclerosis, they are all connected to autoimmune disease. So if you can introduce tolerance, you're in a better place. So that's the end of my talk for now. It's my 10 minutes and looking forward to speaking with you all in a bit. Thank you.
Thank you so much, Professor. That was a great presentation. Next, we will be moving on to the breakout rooms.